this seminar marks the beginning of the second cycle of this RNA collaborative seminar series, which has now expanded to, as I was told, 16 RNA centers across the world. So that's really impressive. Our original moderator, Carl Neugebauer, unfortunately is stuck currently at the airport and cannot join us. So I'm going to introduce our first speaker, Stephen Wang from the Department of Genetics and Cell Biology. Stephen received his bachelor degree from Peking University in 2006 and did his Ph.D. in molecular biology at Princeton University. And after that, he did his postdoc training with Xiaowei Zhuang's lab at Harvard University. For his outstanding work, he has received a long list of very prestigious awards, including the American Physical Society Award for Outstanding Doctoral Thesis Research in Biological Physics. He was Jane Coffin Childs postdoc fellow. He received the International Union of Pure and Applied Physics Young Scientist Prize in Biological Physics. And he's also an NIH Director's New Innovator. And he was also the 2018 35 Innovators Under 35 of China. And there are 700 million people under 35 in China. So how impressive is that? So with that, let's welcome Stephen. And the talk of his title is Image-Based Spatial Genomics and Transcriptomics.
Thank you, Junjie, for the very flattering introduction. And thank you, everybody, for joining this talk. And today, I'm going to talk about our work on the image-based spatial genomics and transcriptomics. And particularly, what we want to introduce about our recent study on multiplex imaging of nucleome architectures in single-cell soft mammalian tissue. And I'm also going to talk about some unpublished analysis using this technique. Our lab is broadly interested in how DNA is spatially organized in a cell nucleus. And we know the interfaced 3D genome is spatially organized. On the first level, we have DNA wrapping around these histones to form these nucleosome structures, and these are also known as 10 nanometer fibers. And on the other end of the spectrum, we have different chromosomes actually occupy distinct nuclear space during the interface of a cell cycle. And these are called chromosome territories. And in recent years, thanks to a very powerful sequencing-based technique called Hi-C, we now know that at the length scale of about several hundred kb, there are these topologically associating domains or TAD structures. So these are continuous sections on the genome that are making a lot of self-interaction. And within TAD, there are often chromatin loops, such as promoter enhancer loops, and sometimes the TAD itself is a giant loop itself. And these TADs are proposed to be important building blocks to build up these chromosome territories. And when they do so, they seem to be sorted into two compartments called the A and B compartments. The A compartment is enriched with active chromatin, and the B compartment is enriched with inactive chromatin. Other sequencing techniques also profiled regions of the genome that are associated with the nuclear laminar or with the nucleolus. And these are termed laminar-associated domains, or LADs, or nucleolus-associated chromatin domains, or NADs. And both of them are associated with the inactive chromatin. However, despite all these beautiful advances, there are still many, many unknowns in the field of 3D genomics, largely due to technical limitations. So there are two conventional traditional techniques that people use to study 3D genome organization, FISH and Hi-C. So both are very powerful techniques. However, both still have some limitations. So just to quickly go through the two techniques, FISH stands for fluorescence in situ hybridization. And in conventional DNA FISH, what we have are these oligonucleotide probes that have the sequence that's complementary to their genomic targets. So these are labeled with fluorescent dyes. And upon heat denaturation, you can denature the genome. And then these probes can hybridize to their genomic targets, and they can be imaged with fluorescence imaging. The challenge of FISH is that because there are only a few spectrally distinguishable fluorescent colors available. So you can only detect a few genomic loci at a time. So this is really not a systematic technique. However, because it's an imaging technique, you can easily see single cells and you get the real 3D positions of all these genomic loci. On the other hand, Hi-C stands for high throughput chromosome conformation capture. It's a biochemistry and sequencing-based techniques to measure the contact frequency between pairs of genomic loci in the whole genome. So it's really a systematic technique to profile the 3D genome organization. However, it measures the contact frequency rather than the real 3D positions. So it doesn't really give you directly the 3D folding path of the chromatin. You kind of have to infer that from the chromatin contact. Also, it usually requires population averaging to get high enough genomic resolution. There are single cell versions of the Hi-C technique available. However, most of the Hi-C discoveries up to this point are still based on population averaging. So we see the key technical challenge here is how do we get real 3D position measurements in single cells at the systematic level, right? So FISH gives you 3D positions and a single cell, but not really that systematic. Hi-C is really systematic, but doesn't directly give you 3D position information and usually require population averaging. And because of this key technical limitation, there is a key biological question that people cannot answer for decades. That is, what is the 3D folding path of chromatin at length scales above the nucleosomes? So from the TADs to the AP compartments to chromosome territory, these are largely basically bigger and bigger chromatin blobs. So how is the chromatin really folded in 3D inside of these structures? To address this question, we basically developed this chromatin tracing technique when I was a postdoc back in Harvard in Xiaowei Zhuang's lab. This is a technique to directly trace the spatial organization of numerous genomic regions in individual chromosomes in single cells. So the idea of chromatin tracing is we want to image numerous genomic loci along the same chromosome, and we want to link them one after another to reveal this chromatin trace. However, if you just label so many genomic loci in a chromosome, one issue is you can't really tell, like they're all there in space and which one should be linked with which one. And another issue is if you label so many genomic loci, in fact, their fluorescence are all gonna be connected to each other. This is due to the diffraction limit of light microscopy. Even a perfect geometric dot, if you image it, it will appear as a disk in fluorescent microscopy. And so we cannot even resolve so many genomic loci. To deal with these issues, we developed this sequential imaging idea. So the idea is we just first label one genomic locus initially. And because we just have one locus, we can actually image it and fit its center position with very high accuracy. And this accuracy is at the nanoscale, and it's much better than the diffraction limit of light microscopy. And then we can photo bleach it and label and image the next genomic locus. We can fit its center position again, and we can link it to the previous genomic locus because we know by design they are next to each other on a genomic map. And we can bleach this second genomic locus and image the third one, really like get its center position with that super high accuracy, link it again. And we do this repeatedly until all the genomic loci of interest are sequentially imaged, pinpoint, super resolved, and we can trace the chromatin folding path of this 3D folding. To realize this design, we developed this sequential fluorescence in situ hybridization technique called chromatin tracing. And here to trace the spatial organization of all 34 TADs in human chromosome 21, we first hybridized to the sample a library of these oligonucleotide probes, and they target the center 100 kb positions of all these 34 TADs. Each of the primary probe is labeled with a fluorescent dye. So at this stage, if you take a 3D fluorescence imaging, you see two copies of fluorescent patches on top of the nuclear background, and these two patches corresponds to the two copies of chromosome 21s in this human lung fibroblast MR90 diploid cell line. However, we cannot see the chromatin traces yet, and after photo bleaching, what we do is we flow in two secondary probes that binds to the overhangs on top of the primary probes that are associated with two TADs only. And these two secondary probes are labeled with distinct fluorescent colors. So upon 3D fluorescence imaging, we see two fluorescent foci that are largely diffraction limited, and we can fit the center position with a nanoscale accuracy again, and get the 3D positions of these two TADs. After photo bleaching, we can flow in two other secondary probes. And so this way, the TADs are basically imaged two at a time until all of them are sequentially imaged and their center positions pinpointed, and the 3D folding path of this copy of chromatin can be revealed in 3D. So in this initial chromatin tracing work, we studied this chromatin folding from TADs to AP compartments to whole chromosome territory in this human cell culture. And we immediately revealed the answers to several biological questions, including, for example, how is the spatial distance of TADs correlated with their contact frequency? How is the spatial distance TADs correlated with, follows their genomic distance? And we disproved some previously very popular models at this length scale. On single copies of a chromosome, we ask, are the AB compartments really stable structures in individual copies of chromosome? Actually, based on previous high-C work, people actually always doubt, are the AB compartments really physical structures that are in individual copies of chromosome like this, or are they just transient trends of pairwise interaction between A regions and B regions, and you only see them due to the population averaging? And with our single chromosome traces, we actually can first find the AB compartment scheme, and we find that in single copies of chromosome, indeed the AB compartments, they are stable structures, and also they are positioned in this polarized side-by-side kind of fashion in a single copy of chromosome. This is actually different from a lot of people's proposals saying that A should wrap around B in single copies of chromosome. And because we not only have single cell resolution, we actually have single copy of chromosome kind of resolution. If two copies of chromosome in the same diploid cell are different, we actually can see that. And we take advantage of that to look at the different chromatin folding between the active and the inactive X chromosome in this human female cell line. And we show that actually the inactive and active X chromosome have quite different folding conformation, not only in terms of that the inactive X chromosome is more compact than the active chromosome, which we previously know, but also in terms of long-range chromatin intermixing, and also this compartmentalization scheme. On these levels, Xi and Xa are drastically different also. And this chromatin tracing technique have led to many following studies from many labs, including us and our collaborators, and it has been applied to shorter genomic length scales, such as the subTAD length scale, the length scale of promoter-enhancer interactions, and also to different model organisms, such as C. elegans and the Drosophila embryos. However, you see that up to this point, we still haven't talked about RNA much yet, right? This is a RNA seminar series, so what's the deal here? In the rest of the talk, I'm gonna focus on a new technique we developed here at Yale called the MINA. So this technique actually combines multiplexed DNA, RNA, and protein imaging, and we use it to profile multiscale chromatin folding, other nuclear architectures such as the laminar and the nucleolus association we talked about, and the numerous RNA species in single cells in mammalian tissue. So you will see the major theme here is we want to have one ring to rule them all, and in the darkness find them. We do this not because we can, but because we have several burning biological questions. One of them is, in the native tissue environment, how is the genome folded in 3D across different length scales in single cells of different types? This question is largely inaccessible to, for example, high-C, even single cell high-C, when you are working with a tissue with mixed cell types. And then how is the 3D genome folded in relation to other nuclear components, such as the laminar and nucleolus associations? So previously, different sequencing techniques are needed to profile the folding and also the association with laminar and also the association with nucleolus. Can we get all these information on the same cells? And then how are the 3D genome variations among the cells cell types associated with gene expression changes, right? So, like in a sequencing world, it would be equivalently as if one is doing like RNA-seq and Hi-C on the same cell. And we try to do this with imaging and answer this question. And also, how are the cell types arranged in space, right? Sequencing techniques often have to dissociate cell from the substrate. So you don't know how the cells are originally positioned in the tissue. And we hope to reveal, are there particular pairing or clustering relationship between the different cell types? And what kind of molecular interactions may underlie the cellular arrangement? So to answer all these questions in MINA, or multiplexed imaging of nucleome architectures, we image a list of cellular and nuclear structures. So first of all, to reveal the multiscale chromatin folding, we imaged first all 50 TADs along mouse chromosome 19. So this gives us the chromatin folding from TAD to compartments to whole chromosome. And then within a TAD, we also traced and imaged 19 consecutive 5 kb regions that are upstream of an important gene that's called SCD2. That's essential for fetal liver development and lipid metabolism. And people previously don't know what's the cis regulatory region interactions for this very important gene. So this actually spans 4 orders of magnitude of genomic length scale in the same cell. And then to detect the cell type specific chromatin folding pattern, we imaged 137 different RNA species at a single molecule level. And 55 of these RNA species are cell type markers. So using those, we can distinguish different cell types in fetal liver. And the rest of them are actually genes on chromosome 19. So we can actually ask what's the association between the gene expression changes versus the chromatin folding changes. And then to study the relationship between the chromatin folding and other nuclear components, we image the whole nucleus and the nucleolus so that we can profile the association of all these chromatin locus with nuclear laminar and the nucleoli. And then because we have, like all of these are done in situ, so we have the cell positions by default, we can study how they are grouped and paired in space and what are the corresponding signaling RNA molecules inside neighboring cells. So I'm gonna quickly go through some raw images and we're gonna go into the biological conclusions. So here is a large scale chromatin folding trace of 50 TADs folded into this whole chromosome territory. This is a single trace of this fine scale chromatin folding of the cis regulatory region of that important SCD2 gene. This is just a very small region showing the whole nucleus labeling and the nucleoli labeling, and we can computationally extract the laminar region and the nucleoli region. And then to image the 137 different RNA species, we actually combined another multiplexed RNA fish technique called the MERFISH that I co-developed when I was a postdoc. So this is also based on highly multiplexed sequential fish. The extra development here is we actually used a combinatorial barcoding and ever robust encoding scheme borrowed from telecommunication so that we can image hundreds to thousands of different RNA species with just tens of rounds of imaging. And to give you just a visual representation, so this is a typical field of view in our fluorescent microscope, and after image analysis, here each single dot is a single copy of an RNA, and the different colors actually corresponds to the 137 different RNA species. And you see that, I mean, so these are from many, many cells. In order to segment them into individual cells, we actually use this lectin WGA to label the cell boundary. And we computationally extracted the cell boundary, and so that we can assign individual RNA molecules into individual cells and get the single cell RNA expression profile. So with the single cell RNA expression profile, we can easily do some, like standard bioinformatic analysis for single cell RNA-seq, using this t-SNE 2D representation and also high dimensional data clustering. We can identify cell clusters that corresponds to different cell types based on the marker gene, right?
And this SCD2 gene that we have talked about was known to be expressed in fetal liver hepatocyte. And from our data, indeed, we recapitulated that. It's highly enriched in fetal liver hepatocyte. So with now the cell identities all identified, we can ask how the 3D folding of chromatin may differ between different cell types. So first, we ask at the short length scale, what is the chromatin folding conformation of the cis-regulatory region of the SCD2 in hepatocyte, right? So upstream of the hepatocyte promoter, there are several clusters of enhancers shown in orange here, and which one or ones of them interact with the SCD2 promoter was unknown. With our data, we can answer that question, and we basically can look into our traces in hepatocytes and non-hepatocytes separately, and we find that, okay, so this region 19 actually shows a much shorter distance with this region 19. So 16 corresponds to one of the enhancers, and 19 is the promoter region, and this interaction shows up in hepatocyte, but not much in non-hepatocyte. And not only the distance is shorter in hepatocyte, we can also count the contact event from individual copies of chromosomes, and we find that indeed the contact is more frequent between these two in hepatocyte over non-hepatocyte also. So we conclude that MINA can actually reveal the fine scale chromatin folding and shows a cell type specific promoter enhancer interaction in fetal liver hepatocytes. Then we look at the larger scale chromatin organization from the TAD to the AB compartment to the whole chromosome. And we ask, what are the cell type specific AB compartmentalization scheme in fetal liver? Using the large scale chromatin traces, we did the compartmentalization analysis and got this compartment score plots. The way to read these plots is, so the x-axis is corresponding to the ID number of the 50 image TADs on mouse chromosome 19. The y-axis is the compartment score. And if you see the red bar, that means it's a positive score. That means that region is compartment A. And if you see the blue bar, that region is compartment B. And we see that basically the compartmentalization scheme between the different cell types differ quite a bit. However, no matter which cell type we look at, when we look at the individual copies of chromosome, the AB compartment are still polarized in this side-by-side fashion, consistent with our previous work in cell culture. And this compartmentalization change is associated with RNA expression change, because we can find when a region is switched from compartment B to A or become more compartment A-like, the corresponding gene in that region typically becomes more active. So we conclude that we can discover cell type specific AB compartmentalization schemes in fetal liver and show their relationship with gene expression changes. Then we look at that. So what is the relationship between the AB compartmentalization and the nucleolar and laminar association? And here we plot the probability for each of the TADs to be close to a nucleolus or to nuclear laminar, and we define this as nucleolar association ratio or laminar association ratio. We plot them against the compartment score. And we find largely there is this negative correlation, meaning compartment B is more associated with nuclear laminar or nucleolus, which is expected based on, inactive chromatin is enriched in all these regions. But we also find there's actually systematic difference between the level of laminar and nucleolar association among different cell types. For example, this pink stuff here is data from erythroblast, and the blue is from pro-erythroblast. These are very closely related cell types. Their AB compartmentalization scheme is almost identical. However, one systematically show higher nucleolar and laminar association ratio than the other. And on the other front, people have been asking me, so Stephen, you see this polarized organization of the AB compartments. Are they due to the B being dragged by laminar or nucleolus? With our new data, we show that's actually not the case, because you can have chromosome that's not associated with laminar or nucleolus, and they show similarly polarized organization between the A and B compartments. So we should conclude that the laminar and nucleolar associations are not solely determined by the AB compartmentalization, nor vice versa. And then we actually found this unexpected localization of genomic loci relative to chromosome surface. In short, what we're calculating here is the probability of a TAD to be on the surface of the chromosome 19 territory. And we found that basically the TADs with very negative compartment score or very positive compartment score are on the surface of the chromosome territory. And these are basically TADs that have very strong compartment A or very strong compartment B identities. And these are actually exactly opposite to what we expected, because using the polymer simulation, we can actually show that based on a phase separation or self-association kind of model, the A and B regions that have very strong A or B identity, they should serve as hubs. They are the most sticky hubs in their phase. They attract all the other TADs with a similar compartment identity. So they should be buried in the center of the chromosome territory. However, like we just see the opposite to what our simulation shows us. And in our simulation, we try to further add extra chromosome interaction. So if we further add A interaction with extra chromosome stuff and B interaction with extra chromosome stuff, we can recapitulate the observed organization. So this shows a balance between the intrachromosomal and the extra chromosomal interactions are necessary to establish the observed organization. And finally, some unpublished analysis, we actually looked into how are the fetal liver cell type arranged in space. So what we have are basically these images, and you can project the identified cell type back onto the images. And we can calculate, are there pairs of cell types that are preferentially neighbors? They're enriched in a neighborhood than statistically expected. So if we do that analysis, we find these enriched neighbors. And the cyan dots here actually are showing statistical significance that's repeatedly recapitulated in each biological replicate. And this chart diagram can shows, like summarizes this pairing kind of a relationship. Some of the pairing relationship was previously known. For example, you see this macrophage in the pairing with erythroblast, and people know that there are these structures called the erythroblastic island. So basically you have a macrophage in the middle and erythroblast surrounding it. And it's known that the macrophage actually regulate the erythroblast differentiation like surrounding it. However, some of the pairing is not well known. For example, we see that there is this pairing between hepatocyte and the pro-erythroblast. And then we basically checked into our merfish data and see like what kind of signaling molecule interaction may be underlying this. And the one pair of signaling molecules called our eye, we see that often this hepatocyte is expressing KIT ligand, and the pro-erythroblast is expressing KIT. So maybe the KIT and KIT ligand interaction is underlying this. And we're further systematically checking into this for all the other cell type pairing relationship we see. All right, so as a summary, chromatin tracing allows direct spatial tracing of the 3D folding paths of chromatin in individual copies of chromosomes in single cells. RNA merfish realizes numerous RNA species in single cells and allows spatial analysis of cellular organization and RNA distribution in tissue. MINA enables multiscale integrative genomic imaging in a complex mammalian tissue and allow de novo discovery of a nucleomic organization associated with gene expression in a cell type specific manner. I'm gonna skip to the biological summary and thank my postdoctoral advisor, Professor Xiaowei Zhang, and my former colleagues at Harvard who contributed to the chromatin tracing and merfish technique, and thank my lab members who contributed to the MINA technique, and my collaborators on the fetal liver work are Dr. Samuel Katz, Dr. Sherman Weisman, and Dr. Qinghua Pan. And we have postdoc positions available, so we welcome applications. Thank you all.
Thank you very much, Stephen.
Thanks.
For anyone who wants to ask a question, feel free to unmute yourself and ask it. Alternatively, you could also type it in the group chat, and I'll read it out loud.
Oh, I see one request asking me to share the paper. Okay, no problem, definitely.
Where do you see that?
In the group chat. Oh, it's privately sent to me, sorry.
Are people able to unmute themselves?
I think we are.
Okay.
This is, okay, maybe I can even. turn my camera on. This is Niels Walter speaking. Great talk. I just had a quick question. Is there any way you can think of to actually look at the dynamics of the TED rearrangements over time?
That's a great question. That's a great question. So up to this point, all our technique is actually on fixed tissue that needs to be fixed and permeabilized. So we basically are taking snapshots of the 3D genome and 3D transcriptome localization. However, other people, I think, for example, Bo Huang's lab at UCSF, they have tried to do the dynamic imaging and this kind of fixed omics level of imaging in a sequence. So basically, they track live cell and all their nucleome behavior in live cells first, and then fix them on the cover slip and do the rest. So this is actually one way to bridge the two.
Thanks.
Thank you.
Stephen, I might have asked you this before. You're imaging these diploid cells. Have you ever looked at the two alleles individually and see how do they compare to each other?
Great question. So one thing we're particularly looking into is actually the XA versus XI, and those are indeed quite different. We tried some analysis like on the initial cell culture MI90 cell line kind of work to compare the two alleles of chromosome 21, chromosome 20, chromosome 22. My impression is I didn't find anything that's super striking, so we didn't talk about that in the paper, but we definitely have that capacity. So if there is a difference, we will be able to see that, like in other settings. Say that again, Junjie.
Like cells with 3 or 4 of the same chromosomes.
Great, indeed, great question. Yeah. So in fact, the liver is during aging there are tetraploid cells that are being accumulated also, and also in cancer there are aneuploidy and all these things happening. And indeed, one biological question is what does the 3D chromatin organization change when that happens? I think that's super interesting.
So Xavier in the chat is asking whether Murfish can provide insights on the role of link RNAs on maintaining the chromosome conformations.
Great question. Yes, I think, yeah, the answer is yes, in short, definitely. I think it's applicable to not just the mRNA, but non-coding RNA as well. And in terms of the regulatory mechanism of chromatin organization, we know actually not just protein plays a role, but also non-coding RNA is also quite important. In fact, in the XI work, we're quite interested in, for example, the exist non-coding RNA, how it affects the chromatin folding. Definitely, yeah, great question.
Okay, if there's no more questions, shall we move on?
All right, great. Okay, I'll take over the host. Let me first stop sharing. Oh, sorry, did I? Okay, all right, great. It's my great pleasure to introduce my colleague, Professor Junjie Guo from the Neuroscience Department of Yale School of Medicine. So Junjie originally came from China, and he received his Bachelor of Art degree from Peking University. So we're actually alumni, although I was in the physics department. And he completed his PhD thesis in the Department of Neuroscience at the Johns Hopkins University School of Medicine, working with Professor Hongjun Song on neuronal DNA methylation in the adult brain. And later, he did his postdoctoral study in Professor David Bartel's lab at the Whitehead Institute. And his lab at Yale is broadly interested in the question at the intersection of RNA biology and neuroscience. And his contributions to the field has been recognized by many very prestigious awards, including the NIH Director's New Innovator Award, the NARSAD Young Investigator Award from the Brain and Behavior Research Foundation, the Klein-Genstein-Simon Fellowship Award in Neuroscience from the Klein-Genstein Fund and the Simon Foundation,
the Yale scholar in neurodegenerative research from Yale School of Medicine, and his postdoctoral work was recognized by the very prestigious Damon Runyon postdoctoral fellowship. When he was a graduate student, he also won like a very prestigious graduate award also, such as the Peter Patricia Cooper International Research Award in Neuroscience and the Harold M. Wendell Graduate Student Award. And with great pleasure, I give the floor to Professor Junjie Guo.
Thank you, Stephen. Can you see my slide?
Yes.
Mouse? Can you see my cursor?
Yes, indeed.
Okay. Yeah, so as many of you have noticed, I've broadened the title of my talk a little bit so that I could share with you some of the emerging work in my lab. So my lab is generally interested in the post-transcriptional regulation, especially translation control of RNAs. Today, I'm going to talk about some of our some of our recently published work, some observations on the global translation and mRNA surveillance deficits in a subtype of ALS-FTD. And then next, I want to talk about some of our ongoing effort in trying to find translation inhibitor for SARS-CoV-2, the coronavirus. Okay, so my lab has been interested in this sort of growing list of human diseases that are caused by the expansion of short tandem repeats, also known as microsatellite sequences, especially in the non-coding parts of the genome. The mutation that we'll focus on today is this hexanucleotide repeat sequence GGGGCC or G4C2 within the first intron of this gene called C9orf72. The expansion of this hexanucleotide repeat sequence from several tandem copies in normal individuals to hundreds and sometimes more than 1,000 tandem copies is the most common cause of inherited forms of not one but two neurodegenerative diseases, including amyotrophic lateral sclerosis or ALS, which involves the degeneration of both upper and lower motor neurons, as well as frontotemporal dementia or FTD, which involves primarily degeneration of cortical neurons. So the mechanism by which this repeat expansion mutation causes the disease has been a very active field of research. Because this is an autosomal dominant mutation, it is often thought that one or more toxic molecules are being produced by the repeat expansion DNA. These toxic molecules can either be the repeat RNAs themselves, including both the sense and antisense repeat containing RNAs, which often form these internuclear inclusion or foci that are thought to sequester a number of RNA binding proteins. These toxic molecules can either also be the polypeptides that somehow gets translated off of these repeat containing RNAs through a poorly understood mechanism known as repeat associated non-AUG translation. And the translation product, these repetitive polypeptides also form these insoluble inclusions that can be found in patient neurons, which are reminiscent of many other neurodegenerative diseases. So the question that I'm going to discuss today mostly has to do with the question why these intronic repeat RNAs can persist and go on and be translated in patient cells. As we all know, once an intron is excised or spliced out from the pre-mRNA, it is very rapidly debranched and degraded inside the nucleus. And in the rare instances where an intron fails to be recognized and therefore being retained in the mRNA and gets exported to the cytoplasm, these so-called retained introns often introduce a premature stop codons to the coding sequence and makes this RNA a very good substrate for an mRNA surveillance mechanism known as nonsense mediated decay or NMD. So the observation that these mRNAs can persist in patients gets translated led us to hypothesize that perhaps there is a global deficit in one or more mRNA surveillance mechanism in these patients. So this hypothesis makes a number of readily testable predictions. One of them is that if there's a global deficit of NMD, then we would see the accumulation of not just this one repeat containing intron in C9orf72, but perhaps many other, if not all other introns in the genome. So to test that, we took the existing RNA-seq data previously obtained from postmortem frontal cortex tissues from either control subjects, sporadic ALS cases that do not carry this repeat expansion mutation, or the ALS cases that are caused by the C9 repeat expansion. And for each subject, we calculate for all the introns the RNA-seq density relative to the read densities from the exons of the same gene. And from this distribution of the intron read densities of all the introns in the transcriptome, we can see that for the sporadic ALS cases that do not carry this mutation, the distribution has a median very close to 0, indicating that the distribution is very similar to control subjects. In contrast, the cases that are caused by the C9 repeat expansion are globally shifted to the right, indicating that indeed there are a very large number of introns that accumulate to much higher abundance compared to controls. In addition to these intron-retaining mRNAs, NMD is also known as a gene regulatory mechanism in that it also degrades a large subset of normal human mRNAs. And similar to these intron-retaining mRNAs, we see that the regulatory targets of NMD are also have higher abundance in C9 ALS patients. And the mRNAs that encode NMD factors are also have higher abundance in C9 subjects, which is consistent with this known autoregulatory feedback of NMD. Central to NMD is an ATP-dependent RNA helicase called UPF1, which also functions in a variety of other RNA decay pathways, including the degradation of these unique mRNAs that have stem loop structures in their 3' end and encode histone proteins. Interestingly, we also see that these canonical stem loop containing histone mRNAs to accumulate to higher levels in C9 patients. So the higher abundance of all these categories of mRNAs led us to believe that there is indeed a global deficit in perhaps not only NMD, but many other RNA decay pathways that involve this helicase UPF1. So to know what exactly the molecular cause of this NMD deficit might be, we ectopically expressed each of the 2 sense and antisense repeat RNAs, as well as each of the dipeptide repeat proteins in HEC293 cells. And after that, we measured the abundance of selected sets of NMD targets that we saw to be more abundant in C9 patients. And interestingly, we see the accumulation of these NMD targets specifically in cells that express each of the 2 dipeptide repeat proteins that contain lots of arginine residues. And those are the polyglycine arginine or poly-GR, and the polyproline arginine or poly-PR peptides encoded by the antisense RNA. So these arginine-rich DPRs have been shown to cause a number of cellular deficits. And we actually found that most of these cellular deficits have very little to do with this NMD deficit phenotype that we observed. But instead, we see that, in consistence with previous work, the expression of these arginine-rich DPRs could significantly reduce global translation, as indicated by this nascent chain labeling experiment using pyrimycin. And we know that NMD is a translation-dependent mRNA surveillance mechanism. If there's global translation repression, then one would expect NMD to be affected as well. So to more accurately quantify the effect, the extent to which the effect of these arginine-rich peptides on NMD could be explained by their effect on global translation, we treated mouse primary cortical neurons with gradients of concentrations of either synthetic poly-PR peptides or cyclohexamide, a translation elongation inhibitor. And at each treatment, we would quantify the level or the extent of global translation repression, as well as the extent of increase in NMD target abundance. And from these largely overlapping dose-response relationships for the PR peptides and cyclohexamide, we could conclude that the effect of these arginine-rich DPRs on NMD can be mostly, if not completely, explained by their effect on global translation. And finally, we tested whether the manipulation of these NMD factors might be able to modify the toxicity phenotype of these arginine-rich DPRs. Inconsistent with previous work, we saw that after 48 hours of treatment of poly-PR, more than 80% of these primary cortical neurons would undergo apoptosis, as indicated by these very obvious beating pattern in all of their processes and cell bodies. However, if we overexpress UPF1 prior to the addition of poly-PR peptides, then we now see a significantly increased survival of these neurons. And this effect appears to be dependent on the NMD function of UPF1 because none of the 4 NMD deficient UPF1 mutant was able to confer similar resistance. I should also mention that very similar results on this neuroprotective phenotype of UPF1 overexpression have recently been reported by several other groups working with Drosophila models that ectopically express these arginine-rich DPRs. So in brief summary, we think that there is this potential positive feedback loop in C9ALS FTD where the production of these arginine-rich dipeptide repeats from the repeat-containing C9orf72 mRNA was able to at least partially inhibit global translation, and as a result, inhibit the translation-dependent mRNA surveillance, including NMD. And the crippling of these mRNA. And I suggest you to read a recently published paper where you can find many other analyses that I don't have time to go into today. So for the rest of the talk, I want to share with you some of our unpublished work on finding a translation inhibitor for SARS-CoV-2 genomic RNA, which is arguably the most important RNA for the past 8 months, and unfortunately still counting. So as most of you have known by this point, so SARS-CoV-2 genome, the SARS-CoV-2 genome is a 30 kb long single-stranded RNA. And most of these so-called non-structural proteins that are critical for the viral transcription and replication are encoded in this super long open reading frame known as ORF1. And the translation of this ORF1 is very interesting in the way that about midpoint of the open reading frame, the ribosome needs to undergo a -1 frame shift before it can continue on and translate this now famous RNA dependence RNA polymerase, as well as a number of other key viral enzymes critical for replication. This -1 frame shifting is promoted by the so-called frame shift stimulating element, which consists of 2 parts. One is this 7 nucleotide long slippery sequence where tRNAs realign in the -1 frame. And the rest of the element is this hypothetical 3-stem pseudoknot, RNA pseudoknot structure that is highly conserved across coronaviruses. And as an example, this pseudonaut structure only differs from SARS-CoV that caused the 2003 outbreak by this one unpaired adenosine nucleotide. And also, any non-frame shifted ribosomes will terminate in this in-frame stop codon that's labeled in red here. And a number of recent studies have focused on this RNA pseudonaut structure and have reported very interesting structural dynamic of this RNA structure. And I highly suggest you to check out these interesting studies. So because of the conservation of this frameshift elements, we think that any small molecule or strategies that could significantly increase or decrease the frameshift efficiency would be presumably deleterious to the virus. So in order to find such small molecules, we did something quite analogous to what others have done for other viral frameshift signals by inserting this frameshift sequence between two fluorescent protein coding sequences with an upstream M-cherry reporting on the overall translation level of this mRNA and a downstream GFP sequence in the -1 frame reporting on the frameshift frequency. And as you can see in these representative image from the cells transfected with this virus, with this frameshift reporter, we see that there is relatively low but robustly detectable GFP signals being produced from this reporter. And in contrast, if we move the GFP back into the 0 frame, then we lose all the GFP signal, which is consistent with the idea that any non-frame-shifted ribosomes will terminate in the in-frame stop codon before it has the chance to translate GFP. So after we teamed up with the Yale High Throughput Center in the West Campus, we treated HEC293 cells with each of more than 4,000 compounds from several libraries containing both FDA-approved drugs as well as several thousand clinical stage drug candidates. And then transfect in our frameshift reporter, and then after 24 hours, image these cells and then quantify the GFP to M-cherry ratio. And as you can see from this effect size distribution, the vast majority of these more than 4,000 compounds have very little, if any, effect on the efficiency of frameshift. So out of the several selected compounds that have the highest effect size, we reordered these compounds and tried to validate them using an orthogonal luciferase-based frameshift reporter assay. And out of these 8 compounds, we were able to validate a weak frameshift enhancer, which is interestingly a drug that is currently under clinical trial for COVID-19, as well as a relatively robust frameshift inhibitor, which we'll focus on for the rest of the talk. So this compound, CI934, belongs to a very large family of antibacterial compounds that are known to target bacterial DNA replication machinery. But interestingly, the compound libraries that we used also contained at least 40 other analogs in this compound family, but none of them has nearly as much of an effect as CI934. Suggesting that this activity might be a novel activity of this compound. And from the dose response, we could see very nice and progressive inhibition of -1 frameshift as we treat cells with higher concentration of this compound, with around about 50% inhibition of -1 frameshift seen at around 20 micromolar of this compound. And at the highest concentration of this compound, we could inhibit -1 frameshift by more than 90%. And as expected from the extreme sequence similarity that I already mentioned between the frameshift sequence between SARS-CoV and SARS-CoV-2, we see almost identical dose response relationship, almost identical effect of this compound. So we went on and tested the effect of this compound on a panel of viral and cellular -1 frameshift sequences and saw that, as I mentioned, the compound was relatively effective on SARS-CoV and SARS-CoV-2 -1 frameshift, which are both beta coronaviruses. Interestingly, it is nearly as effective on the other human common beta coronaviruses, OC43 and HKU1, and less effective on the other coronaviruses, including 2 human alpha coronaviruses, as well as a gamma coronavirus. And finally, we see no inhibition of -1 frameshift in the other viral frameshift sequences that we tested, as well as the endogenous human PEC10 mRNA -1 frameshift. So with the ability of this compound to inhibit -1 frameshift at varying extent, the question is, at what point do we see antiviral activity? By quantifying the cytopathic effect of SARS-CoV-2 in vero cells, we see that the cytopathic effects caused by SARS-CoV-2 was almost completely blocked at a relatively high concentration of this compound that corresponds to more than 80% inhibition of -1 frameshift in our reporter assay, suggesting that a minimum of 20% residual frameshift efficiency might be required for the virus to continue to replicate in vitro. So currently we're collaborating with Brett Lindenbach from the Department of Microbial Pathogenesis to better quantify the activity of this compound on the replication of SARS-CoV-2 as well as other coronaviruses. So to briefly summarize, we performed our high throughput chemical screens, identified this antibacterial compound as an inhibitor for the -1 frameshift of SARS-CoV-2. And this inhibition seems to be quite viral frameshift sequence specific, and it's most effective for beta coronaviruses. And I don't have time to show you the data, but we also find that the inhibition by this compound on -1 frameshift is quite robust against point mutations within the frameshift sequence, within the frameshift elements, including both existing mutations that's already in the population, as well as newly introduced synonymous mutations in this region. And importantly, we think that at or above 80% of -1 frameshift inhibition would lead to very potent antiviral activity, at least in vitro. So currently, we're very interested in further optimize this compound by analyzing the structural activity relationship, as well as trying to understand the mechanism by which this compound can inhibit -1 frameshift in coronaviruses. So both of these 2 projects I described, I talked about today, are led by a very talented postdoc, Yu Sun, with the help of several other lab members, including Aziz, Atta, and Jeffrey, who contributed to computational analysis of the NMD projects. And we're currently working with a number of different labs in trying to better understand this compound and perhaps find additional compounds that with similar activities. And thanks to our funding agencies, we also have postdoctoral positions in the lab. With that, I'd be happy to answer any questions. And thank you for listening.
Great talk, great talk.
Hi, Junjie, great talk. Both projects are very exciting. I wonder, in the targeting of the transcript with retained introns by the nonsense-mediated decay pathway, whether your UPF1 function is dependent on the exon junction complex or depends on UPF2 and 3 or not?
Presumably for the C9 RNA, it is most likely exon junction complex dependent because that's the first intron in the gene, which obviously followed by all the downstream exon junction complex. But I think there is still some question whether the C9 repeat containing intron RNA is an NMD target, even though that was our motivating question. We're still investigating whether that is the case.
Great. I definitely see more questions in the chat. Let me try to read these. All right. So Tatsusaki Kurosaki is asking, he's saying that thank you for interesting talk. You explained that NMD inhibition in C9 ALS is due to global translation repression. Do you know the mechanism why arginine-rich dipeptide repeats block translation?
Yeah, that's a great question. I think there are a few previous studies that have shed light on this question. The inhibition on global translation might happen at multiple levels. It has been shown that these highly positively charged peptides might interact with the exit channel of the ribosome. So that would presumably be one way of interfering with translation. And these highly positively charged peptides could also very strongly interact with the RNA, which again could act as roadblocks for translation. And additionally, we and others have seen that these arginine-rich peptides are very strong binders of nucleoli, which presumably would also disrupt ribosome biogenesis. Although in our short time scale experiments, we believe the translation inhibition is largely unrelated to ribosome biogenesis.
Great. The next question from P. Yao is, what could be the molecular mechanism by which arginine-rich peptide inhibit global translation? I guess it's the kind of the same question. Will this translation silencing effects play a critical role in the pathogenesis in addition to NMD inhibition, which is downstream of the translation changes?
Yeah, that's also a great question. We do believe that NMD is not the only mRNA surveillance mechanism that's affected once translation is inhibited. Presumably, the other translation-independent mRNA surveillance mechanisms would also be affected, although we have not sort of comprehensively tested each one of them.
Great. And a third question.
At least the histone mRNA decays. Histone mRNA decay was also affected.
All right. Another question from Ambro is, I was wondering whether you have made mutations in SARS-CoV-2 to see whether mutations that inhibit frame shifting by 80% also reduce pathogenicity.
We have not done so, but that's a fantastic experiment. Yeah. So there are a few existing mutations that's already being documented in these SARS-CoV-2 genome sequencing global efforts. And so far, I think the largest effect we've seen in our reporter assay after we introduced these existing mutations is about 50%. So that's somewhat consistent with this idea that the virus can tolerate 50% inhibition of frame shift. But it will obviously, like you suggested, be very interesting to test a whole series of mutations that affect frame shift at varying degrees. But we think that our drug provides a way, an alternative way to answer the same question.
All right, so that's the last question in the chat. If nobody else would like to unmute themselves and ask more questions, I guess this wraps up the day. And thank you everyone for joining this day of talks, and we hope to see you next week. Thank you.